It's the Bird Emergency, the show where we focus on all of the problems confronting birds and the marvellous people and the programs that are trying to reduce all of the problems that they face. Climate change, habitat destruction, disease. It never ends. I'm Grant Williams. I'm the host and producer of this show. And today we're going to go into disease. How disease can be an existential threat to birds. The main focus of this show is to encourage you not only to think about the plight of birds and to think about doing something, but to get out, get off your couch, get out, do something. Write a letter, join a group, take part in a citizen science project, do something. It's time to put on your white coat. I have to put on my white coat because we're going to do some hard science. I think it's pretty hard science because I had to struggle to get a grip of this. But as you would know, I'm concerned about the threats to birds. And the Beak and Feather Disease virus is potentially an existential threat to the parrots of the world and particularly the parrots of Australia. Now, Dr. Johanne Martens is speaking to me now, and she's conducted, as part of her PhD work through Deakin University, about a 4-year study into this disease, and she's going to tell us all about it now. Thanks for coming on the Bird Emergency.
No worries. Very happy to be here.
I tracked you down because I saw an article in our one of our major daily newspapers in Melbourne, in Australia, and I don't know if it was a clickbait sub-editor headline, but it was disease and extinction threat for Australian parrots. How could I let that go? How about you tell us what the disease is, and was that an overblown headline? Is it really potentially that serious?
I think we need to do a lot more research because there's just so many unknowns about it still. But the facts are that so far all parrot species worldwide are considered susceptible to the disease or to the virus that causes it, Beak and Feather Disease virus or BFDV. And as you know, there are almost 400 parrot species in the world, and it's one of the most threatened groups of birds in the world as well, with about 30% of the species being considered threatened. So a virus that can potentially affect all of these species can be quite a serious issue. And BFDV has recently been found in non-citizens in Australia as well, so in other bird species. It can jump to new hosts, so it's just important to find out more about it before big outbreaks happen.
If a bird has the virus, what happens to the bird?
It depends a bit on the species. Some symptoms we can see or signs of disease: feather loss, which happens gradually, usually during subsequent molts. The feathers can't regrow properly, so the birds can actually become naked over time. The beak can become really deformed because it's not growing properly, which means that it can make the birds unable to eat. Then they can get tissue damage, internal organ damage. The immune system gets suppressed because the virus replicates in some organs that are necessary for a functioning immune system. So they actually often die of secondary other infections with other pathogens. There are very acute forms of infection, especially in nestlings, where they die within days without developing many signs. But then other species can carry the virus or be infected without showing any signs. So it's really variable and complex, really.
So that raises a couple of questions. Do you have any idea what the mortality rate is of the virus, or does it have other effects, like just making an infected individual, less likely to successfully breed? And is it able to be passed on in a hereditary fashion, or is it infected birds being in contact?
It's a bit difficult to study mortality, especially in wild birds. In captive birds, pet birds, we know that it causes fatalities. In wild birds, we don't know what really happens to birds we don't find again, whether they've dispersed or died. So this is hard to say. It is a bit problematic that the virus can get passed on in many different ways. It can be transmitted directly from bird to bird, for example, through crop secretions. So when a bird feeds another bird, and also through contaminated feather dust and feces as well, which can, for example, also become a problem in aviaries with pet birds or in captive breeding programs for conservation, where these things tend to sometimes build up, especially feather dust, for example. And it can also be transmitted from mother into the egg. So the embryos can get it already from the mother. And what's more, the virus is also very resistant outside the host. A thing that at the moment gets discussed with COVID as well, for example, that a virus can sometimes survive for days or months, maybe, outside the host on surfaces. So one study we did actually looked into that, whether there's a risk of transmission via contaminated nest boxes or nest hollows.
We'll talk about the species that you studied, and we'll go through the sort of the methodology of the study, because I found that really interesting. But we're talking about captive breeding programs just then, birds in captivity, and the article that I saw mentioned the orange-bellied parrot. Has it been in our captive insurance populations already?
It has in the past, yeah. So Beak and Feather Disease virus has been a problem in orange-bellied parrots in the past, and also it's been found in swift parrots as well, our other critically endangered species here in Victoria. So yeah, it can become a problem. Important factors here are that in captive breeding programs, of course, you have birds more closely together, and also you already have a really small remaining population, so you don't have a margin for any fatalities and any losses. And endangered species have not such a good immune response very often due to low genetic diversity, which makes them more susceptible.
I'm not sure whether this is something that you'd be aware of, because it certainly wasn't part of your study, but for a management regime with a captive population, how can you effectively isolate the birds? I mean, do you have to take extreme measures like euthanizing infected bird in an insurance population, or can they, if you've got a complex of maybe a dozen flight aviaries, could you maintain separation with an empty flight in between, or would you need to have two or three? I'm just wondering how containment or how control would affect management. And I suppose it's really sort of interesting because we're in that time in the human population at the moment where we're struggling with a new virus that we don't know much about, and how to manage it and how to continue on with it still circulating broadly in the population. Can you offer any thoughts about that?
So I don't know the details of what they really do in the management so I don't know the details about how they handle it in the captive insurance populations. But it will be hard. From what I've known, they've done a terrific job with the orange-bellied parrot. They have really good hygiene measures in place. But it is a really tricky virus for any captive breeding and recovery program because it is so resistant and stable and can be transmitted via so many different pathways. So it's risky to even have several aviaries in between, in my opinion, because you carry a bit of feather dust from one aviary to the other, and then you have contamination already. If that's enough to infect a bird is another question, and I don't know the details from these programs. But one really important thing to do is to just work in a really, really clean way in any recovery program with nest boxes and so on and so forth. Disinfect with the right disinfectants. Not all disinfectants work against BFDV, which adds a bit of fun to that. And just to be very, very clean and careful. Same as with our current virus situation that you were mentioning.
Let's talk about your particular study. It took about 4 years, and you were studying one of the favourite species for so many birdwatchers in southeast Australia, the Crimson Rosella. Well, how did you decide to do the study, and how did you go through the process of designing what would be an effective way to study the virus?
I was really lucky with the study because I actually came into the project as a PhD student when the study was already set up, and then helped with planning the details. But in my research group with my supervisor, Andy Bennett, they've been doing work on Crimson Rosellas for many years now. Really cool projects on disease, coloration, odor. So I was very lucky in a sense that there were already a lot of nest boxes being put up that I could use for my studies, that we already knew how to trap the birds. So that was really ideal. And then my project is part of a big ARC linkage grant to tackle some of the knowledge gaps with BFDV. And a lot of thought went into, okay, what are the knowledge gaps? What do we still need to find out? And then, look at the literature, the existing literature, and speak with the experts to see how we can best go about analyzing that.
How much was known specifically about the prevalence of the disease and how it was circulating in wild populations in Australia before you picked up your study?
There were quite a few studies from Australia, but most of them were on captive birds and not many at all on wild populations. Another study came out while I was doing my PhD on wild cockatoos, but most other studies were really from birds that were either admitted to vet hospitals, so that were already sick, or from experimental populations. Major piece of work was another PhD thesis, also from my research group, from a previous PhD student, Justin Eastwood, who did a lot of the initial work with BFDV and Crimson Rosellas, analyzing Crimson Rosellas, analyzing the whole species complex of Crimson Rosellas with the Adelaide Rosellas and the Western Slopes hybrids. It's a really interesting study system to look at evolutionary and geographic differences. Whereas I then focused more on the ecological aspect, just in the Crimson Rosellas around Geelong.
Okay, let's talk about the nest box method of keeping an eye on the breeding population and how you trap them, what you did, how you determined that virus was present and what it was doing, because this is where the lab coat stuff really comes in. You've actually probably got your climbing ropes and your ladder, and then you have to go and put your lab coat on.
Exactly. The project had a bit of everything, really. So it was a steep learning curve for me. I worked on urban parrots before moving to Australia for my PhD. So I knew more about the parrot side of things, although I had some lab experience. But it was a really interesting project overall to learn a lot of new things. So as I mentioned before, we already had a lot of nest boxes in place. And me and my colleague Helena then installed another 80 or so parrot nest boxes. So we had a lot of fun looking for the right sites to put up nest boxes and speaking with the landowners. The landowners were all fantastic and helped us a lot in our work. Really good collaborations there. And then we started getting all the nest boxes ready. So we had about 250 nest boxes in total for Rosellas. And then just before the breeding season, we got them all ready, repaired them, then started monitoring them. So went there, I think in the beginning we went every week to check what's in the boxes, to then record numbers of eggs, numbers of chicks, laying dates, hatching dates, to estimate the age of the chicks. Once the chicks were hatched, we started catching the parents. We have a special setup where we can shut the birds into the nest boxes when they come in to feed. Luckily, Crimson Rosellas are extremely tolerant of that. You couldn't do that with any birds, with other bird species. But the Rosellas are great. Once the chicks are there, you can trap them, take samples from them, and then they just go back to feeding their chicks. And we also took samples from the chicks themselves at different ages to see how the prevalence in the nestlings looks like at certain age stages. So we sampled from the fairly small baby parrots when they were about 1 week old, and then again just before fledging at about 4 weeks. So they fledge when they're about 5 weeks old. And during the breeding season and before and after, we also took swab samples of the nest boxes themselves. So we just, we had these little cotton bud swabs, but like the cotton buds you screen your ears, and we swabbed the nest material, and just to see if the virus is also present in the nest material and how long we can detect it for after the breeding season, and because the hypothesis from other studies was that it may stay present in the nest box until the next breeding season, then being able to infect the birds that then go back into the box to breed, because they very often use the same nest sites over and over again. And then in between the breeding seasons, we kept trapping not only Crimson Rosellas, but also other abundant species like galahs, sulfur-crested cockatoos, in walk-in traps. So these are just cage traps that we fill with bait, so seed, apples, and then trap the birds in there, and again take blood samples and swab samples from them, and then release them again as quickly as possible, of course, to cause the least amount of stress. And then we take all these samples back to the lab, and then it's white lab coat time, and the screening starts, which is a lot of manual labor. We first had to extract the DNA from all of the samples, which takes quite a while because we had hundreds and hundreds of samples. We then take the DNA from the blood samples to sex the birds so that we know whether we are testing males or females, because we also wanted to look at the prevalence differences between the males and females as well. And then I screened the blood and cloacal swab samples, so the DNA, for presence of viral DNA and also amount of viral DNA. So that gives us an idea about intensity of infection.
So for the non-techie, non-science listener, and I'm putting myself squarely in that bracket, the swab samples are from the vent region, or are you also testing regurgitated foods? Like, what exactly were the swabs apart from the contents of the nest box?
They were initially we collected oral and cloacal swabs, so from the cloaca and from the beak. I think those oral swabs were the most fun for the birds, especially the cockatoos. Cost us a lot of swabs because they just snapped them in half. So it's quite interesting to get a swab in and out of a parrot beak sometimes. And then I've mainly focused on cloacal swabs to get a bit of an estimate on viral shedding. So are they excreting the virus via the cloaca? Whereas testing the blood samples gives more of an overview of whether virus is present in the body, in the blood. But those birds that have it in the blood may not actually be excreting virus. They may just be carrying it.
So I read that you were measuring, monitoring the weight of the birds in the breeding season. What did that lead you to decide? Like, what were your results from that portion of the study, and what do they mean?
We actually so when we analyzed the weight, we recorded the weight and also combined that with tarsus measurements. So we measured the length of the foot to account for differences in size. So we had body weight corrected for bird size to see if there's an effect of infection on body weight, so pretty much on the bird's condition. So body weight was one way to do that, and then we also measured another blood index, where you measure the ratio of red blood cells and white blood cells in serum. Red blood cells in serum, and these are both just measures for how well the bird is and the bird's body condition. And we didn't really find an effect of infection on body weight or hematocrit. And the only thing we found was that male Crimson Rosellas are a fair bit lighter after the breeding season, which may just be due to the efforts of of breeding, but there wasn't really much of an effect of infection.
What was there any sort of results you could, or conclusions you could draw about the effect of recruitment per pair or per clutch that is affected by if one or both of the parents is infected, or and we're able to compare that to maybe a pair that aren't infected? Does the success rate of each clutch be affected by infection with the virus? That's a really complicated way of saying, but does it affect the recruitment into the wild population severely, I guess, is what I want to know.
Look, that's something that I really wanted to look at. That was one of my objectives, but in the end, I couldn't, due to a really surprising result, which was that the prevalence in breeding birds and the nestlings was super, super low. I kind of had expected it to be the other way around, but yeah, breeding birds hardly had any BFDV infection. So I screened parrots from over 80 nest boxes. So together with the nestlings, that's several hundred samples. And we then had a total of 11 nests where there was either one parent or one nestling infected. So the sample size is then so small. So half of the 11, I think 6 of the 11 had an infected parent, 5 had an infected nestling. So then the sample sizes get so small that you can't really draw any conclusions.
But does that lead potential good news that in the other nest boxes there was no infection?
Absolutely, yeah. And it's really surprising, and I really like to understand better why that happens. And I think we need to do more studies like this to see if that's the case as well in other populations and other species, because all we can say is during my study, this was the case in Crimson Rosellas in Victoria. But other species may deal with it in a different way. But for our population, that seems to be really good news because the prevalence was so low. What was then super interesting, though, was that the young birds, after leaving the nest, had the highest prevalence. So we're trying to find out where exactly do they get infected, and that's still a bit of an open question because we had this super low prevalence. It was something ridiculous, like 1.8% prevalence in the nestlings. And then in sub-adult Crimson Rosellas that are less than 1 year old, we get prevalences up to 80%.
So that might indicate that the virus is extremely virulent at a particular stage of development for the birds, and that the habit of the birds of flocking together when they are sub-adult and they've begun the process of leaving the parental pair, that might aid in the spread and the concentration of the virus.
That's our hypothesis at the moment, or our assumption from the data we had, because parrots are so sociable. They flock together, which of course facilitates the transmission of any pathogen. And young Crimson Rosellas, they hang out with their parents for a while and still get fed after fledging, but then over winter they get together in these foraging flocks that you may have seen as well, where you suddenly see a whole bunch of these young green Rosellas. So the great thing that I forgot to mention about Crimson Rosellas is you can age them really well because they're green up to when they're 1 year old, and then they start turning red. And then they also have a stripe on the underside of their wing. So we can age them pretty accurately until they're about 3, which is great and really unusual for parrots. And so they get together in these big flocks and roam around, and that probably makes it fairly easy to pick something up. And what's more, in young or very old individuals in general, all over the animal kingdom, the immune system is not at its best. So especially young individuals, their immune system is what we call pretty naive. So it hasn't encountered a lot yet. It's not fully developed yet. So that makes them particularly susceptible as well. So they flock together, they're fairly susceptible, and that's probably where these infections happen.
And I'm guessing there's no way to know whether the virus is present, in sort of waiting in those feeding grounds, in those foraging grounds, in litter, in debris, in discarded seed pods, a whole range of things. So I'm guessing that's something that needs to be studied in the future. Now, for the non-scientific community listeners, it's clear that studies like these need to be replicated, not only across Australia, but across parrot populations in the world. I don't know if I'm asking you to let out a secret, but what if someone was to want to replicate your study? How much money would they need to undertake that?
That is so highly dependent on what exactly you want to do and what resources you already have that that's pretty impossible to answer. There are so many ways of doing it. It depends on the sample type you want to test. It depends on the number of samples, the testing assay you want to use. It's hard to say, but we definitely need money for conservation projects to do these things. I think what a lot of the conservation projects do is they send the samples off to commercial labs. So I had the opportunity of doing all of the lab work myself, but there are actually commercial labs that accept samples and then just send you back the results. This is also, for example, how pet birds get tested when you bring them to the vet. The vet takes a blood sample, sends it to the commercial lab. So yeah, it really depends on the sample size and what you're actually planning to do, but it can get quite expensive depending on how you set it up.
Yeah, I think people. I'm certainly interested just so that you can compare how money is spent in the community generally. So yours was a 4 year study, and it's in terms of results and as a management tool, it's a partial study because there needs to be more happen for us to have a complete picture of what's going on. So for a 4 year study like yours, let's assume the methodology was almost exactly the same and somebody in another location had access to the material, the equipment, friendly landholders who were all helping, so a whole lot of uncosted assistance. Would 200,000 cover it, or would that not be enough, or would that be ample?
I can't really tell you. It depends on how much you yourself would get paid as a wage to do it as well. It's quite complex, but $200,000 will get you a long, long way in terms of just cost for sample analysis. I
Okay,
think
and then
because
and then there's a fieldwork component on top of that, and the nest boxes and whatnot. So let
yeah,
so we're probably looking upwards of 300,000 for a 4 year study.
possibly,
Yeah,
yeah. It really depends on how much is already in place.
yeah,
For example, what's a great opportunity is when people are already catching and banding parrots, or already do nest box monitoring for endangered parrots or cockatoos. Then it's once you have the ethics in place, fairly easy to take a few samples while you're already up at the nest box and send them in somewhere. And that's what a lot of studies do. If you have to start buying the ladders, buying a ute, putting up the nest boxes that you've also bought, etc., then it gets expensive. Yeah.
there must be a lot of frustration amongst researchers because when there's a lot of studies happening concurrently, there can be opportunities to collaborate and to reduce costs for everybody. But because we do so few comprehensive studies, those opportunities are missing, and that's a huge opportunity cost. And I'm almost 100% sure that when projects are reviewed or looked on for, or grant submissions are being reviewed, yeah, I doubt whether opportunity cost is ever factored in, but it should be, because it is in all sorts of other projects and spending in government and in business that is easy to measure, but we just don't. So, I would like you to tell me what you think the useful information that's come out of your study is. What people have to make decisions based on what comes out of studies. So I guess there's scientific decisions and there's management decisions, and there's clinical decisions probably that flow from your work, both for managers of wild populations and managers of captive populations. What do you think?
Well, I hope that my results can be useful, definitely. I've got a strong conservation focus myself and would love for the results to be helpful in some way. So I guess the main findings were that I found BFDV in most of the common parrot and cockatoo species I investigated. And interestingly, in absence of clinical signs. So all the birds looked healthy, but I found it in 6 of the 7 species I tested. I also tested blue-winged parrots, which was a bit of a special case. They're really similar to orange-bellied parrots, our critically endangered birds here. And I wanted to find out whether blue-winged parrots also carry beak and feather disease. And that was the case. And beak and feather disease virus is thought to often affect endangered species, or be able to mainly affect endangered species by so-called spillover infections from common species. So it's actually quite a big thing that we do find the virus in so many common species, and that really shows that we just have to be extra careful to particularly protect our rare birds from exposure to that. Another interesting finding was that we did pick the virus up in nest boxes, so in the nest material itself, not just in the birds. And we didn't pick it up as long as we thought we would for such a long time span. So it may not be as persistent in the nest box as previously thought, which is good news. Although, again, to be taken with care, because it may be different between study populations and species and so on and so forth. But at the moment, it looks less likely than previously thought that the birds get infected via contaminated nest material from one breeding season to the next. We did pick the virus up in a lot of nest boxes where we knew parents were shedding the virus, so it could become a risk to some species. We also found that the virus can stay in birds for quite a long time. So we did the study where we captured Crimson Rosellas several times. And so we could have a really good look at the patterns of infection over time, which is quite rare in wild birds. Usually you catch a bird once and then you never see it again. But because we had such a large sample size, we had some birds that we could test several times. And we could see that some birds really stay infected for several months. So that, again, has implications for the fitness of the population when there are a lot of birds that are infected for a long time. And it also shows that they may be able to transmit the virus for a long time without dying themselves from the virus. So all these results hopefully can help in conservation. And the last important finding was that we found beak and feather disease virus year-round in our populations. So whenever we tested, it was there, with some seasonal variation, and with the highest prevalence after the breeding season, which may be something like a high-risk time when all these young birds are becoming infected after leaving the nest.
Now, there's no vaccine available, so there needs to be work on a vaccine I would assume, but then there's the problem of how would you deliver a vaccine to wild populations of birds? Does anyone have any idea how an effective delivery system might look?
I'm not involved in developing a vaccine. One of my colleagues is working on that, and he has been working on it for a long time. It's a really complex, complicated process, which takes a long time, and which has to comply, of course, with ethics and so on and so forth. I don't think we will be able to implement that to wild populations, even if we get the vaccine. I think that'll always be more of a vaccine to apply to endangered species in captive breeding programs, for example, because there it's easy. You vaccinate all the birds in your aviary, and then you can release them or breed with them, and they're safe. But I don't think it's feasible or possible to vaccinate our wild birds.
Yeah, I couldn't imagine a delivery system that could be designed that would work. I mean, especially if it's a intramuscular or intravenous injection that was required, it would seem completely just no chance, just too big, a project that would just be too big. Is there anything else that people need to know about the work you did? I mean, I think it's not too big a statement to say that the virus is potentially an existential threat, certainly to some species. I think that's probably a fair thing. So, what should the public in general know about the disease? Oh, and also, should people be reporting findings if they see affected birds? Should they be letting someone know? Is there a sort of citizen science project that wants to collect that information?
I don't think there's a citizen science project in place. I'm definitely not aware of one. There may be one that I've overlooked. One important thing to do is just to keep bird feeders or water baths, if you have them, really, really clean to reduce the risk of transmission. If you see a sick bird coming into your bird bath, maybe stop supplying, especially at feeders. I think that's easier because that prevents the spread as well through reduced contact of that sick-looking bird with other birds that just all accumulate at the feeder. And it's really important to clean feeders and water baths pretty much daily and replace old seed, etc., to prevent the virus and also other diseases, other pathogens from building up. Another important thing that we can all do is to become a member of the great nature conservancies that we have. Zoos Victoria, BirdLife Australia, they all do fantastic conservation work and need our support.
And internationally, you will find similar bodies, of course. So if you want to do something positive, look in your geographical area for the appropriate body. Now, something just sprung to mind about you mentioning feeders and bird baths, but what about where, cockatoos come in for feeding and might sit on people's railings and come on tables and things like that? Do we have to think that should it become normal practice to treat the surfaces that visiting birds, especially parrots, are alighting on the same way we do in the COVID-19 world, like we do with door handles and buttons in lifts and things like that? Is that something that needs to become commonplace for people who are encouraging wild birds to visit their homes, their gardens?
I think ideally that's what should happen. I know that it's difficult in some cases, but it's not just Beak and Feather Disease virus, which is a big risk for our parrots and can make them suffer a lot if they get it. But there are other pathogens around as well, zoonotic pathogens that can jump between parrots and humans as well. So it's better to just be careful and clean. And while still enjoying our parrots, of course, and interacting with them, but just be very, very aware that there are certain risks to that as well.
So there's a very practical use for those 99.9% effective bacterial and wipes that you have hoarded in your panic buying during the COVID-19 era. Don't let them go to waste. Wipe down everywhere that the birds are alighting and are hanging out, and everything that they chew, and yeah, just do something positive. That's always the message.
Absolutely. Ideally, I mean, even bleach. It sounds a bit brutal, but that's the most effective thing. Let it evaporate properly because before you use your feeders again, so that it doesn't smell of bleach anymore. But that's the most effective thing. Things like BFDV, they are not too fussed about ethanol in these things, so you need harder stuff than that sometimes. But any cleaning will help you and the birds.
That makes me think that for people who are really committed bird feeders and garden bird watchers interacting with their birds, you probably need to be swapping in and out a number of feeders and bathing implements so that you can effectively clean them, but still have the food sources or the water available to the birds who have become used to it and are relying on it. So yeah, go and that can be your economic stimulus. When you get your next bit of economic stimulus, go and replicate your bird feeders and your bird baths and keep them clean.
Or at least invest in a good brush and just scrub them down
Yeah,
as often as possible. Yeah.
exactly. Thanks, Mr. Morrison. That's an Australian joke there. Um, now, yeah, Hannah, that's I think this is going to be like the building block of some really important work in the future. So good on you for sticking with it. And have you actually got your PhD award yet?
No, I haven't yet. My thesis is still under examination, so it's still interesting times. So we'll have to see about the outcome. So I handed it in a couple of months ago, but these things always take quite a long while.
So nearly Dr. Martens. I think that's what we'll have to call you for the moment. Is nearly Dr. Martens. You mentioned that you came to Australia to undertake the PhD. Is that right?
Yeah, that's correct. Yeah.
Okay, so give us the history, the academic history, and the fieldwork history prior to arriving here in Australia.
Oh wow, that was probably pretty much anything bird related. Um, so just before coming to Australia, I was working for the Natural History Museum in Stuttgart in southwestern Germany, studying introduced urban Amazon parrots. I started that work as my bachelor thesis actually a few years before that, and I just kept going with it in my own time because I just find that flock very fascinating and how they can survive in Germany, and then was able to continue that during my employment at the museum. And that was a lot of fun. So Stuttgart, as a German city, has this European flock of neotropical South American Amazon parrots, about 60 of them. They've been around since the 1980s. And when I started to work on them for my bachelor's, there was almost nothing known about them. There was the occasional roost count, but apart from that, people didn't really do anything with them because they were just there. They're introduced. They don't really belong there. So nobody really knew how they survived. And the very first time I saw them as an undergrad student was when someone took me to their roost site. And that was such a bizarre experience that they just stuck with me. So they sleep in the middle of the city, next to the tram, next to the traffic, with cafes underneath. And I stood there, it was getting dark, it was snowing heavily. All the branches were covered in snow, and suddenly these big green parrots come screaming down from the sky and settle in and fall asleep. And I just couldn't believe it. So I worked on them for about, I don't know, maybe 5 years. And we found out a lot about how they breed, how their breeding success is going, their mortality, what they eat, and it was a lot of fun.
Sounds like it. Uh, Yeah, it's amazing that so many of the Amazons and the sort of Indian ringneck complex of parrots have escaped around the world and have set up these crazy colonies and have really dominated local parks and urban streetscapes in so many cities. It's quite freaky. We've got a similar situation, I think, here with the way the rainbow lorikeet has just exploded and dominated in the in the trees and the canopies and the skyline in Australia. They're just everywhere. Well, Eastern Australia, I should say. Perth's got them too now. So yeah. So you, how did it come about that you decided to come to Australia? What's that process like? Like, did you undertake a master's before coming here?
I did, where I did something completely different, bird related, at the Max Planck Institute, um, where we looked at heart rate and body temperature in ducks, actually, with a new type of data logger that just records these physiological measurements in real time of the birds. And it is a bit related because these results can be used for studies on avian influenza or other diseases where you can check in wild birds remotely if they pretty much have a fever, if they're sick, if there's a new pandemic coming, pretty much as a bit of an early warning system. And then I went back to the Natural History Museum afterwards, went back to working with the Amazons, which was great because I have a strong conservation focus and I just find it fascinating how some species adapt to cities, like you also mentioned with the rainbow lorikeets, and others just don't manage that. And I wanted to understand more what affects birds and especially parrots in our very human modified landscapes. Because disease, like I'm now studying, is just one of many factors that putting species at risk that are already under so much pressure from other directions. Habitat loss, poaching is a big thing in parrots. So there are so many pressures from so many different angles. But yeah, I'm digressing on my favorite topic. But so how did I get to Australia? So I was working with the Amazons, and then the PhD position here actually came up as an online advert, and I got that forwarded from several friends of colleagues who said, "Hey, you're working with parrots. You'll find that interesting." And I applied last minute, pretty much 5 minutes to midnight, not sure whether it's a good idea to move to the other side of the world, but being completely fascinated by being able to potentially doing a lot of field work with parrots in Australia, which is so cool, and doing something that's relevant to their conservation as well. So I just applied and was very lucky that my supervisors took me.
So now that you've managed to spend 4 years here working with the very affable Crimson Rosella, I think they they seem like they'd be the good bloke of your local bird community.
They bite though when you have them in hand. Gee.
Is there a parrot that doesn't? I mean, even the Burke parrot loves to give you a big nip, and they're as docile as can be. Uh,
Yeah,
uh,
whenever I help out with songbirds, I'm like, oh, this is different. I don't need leather gloves, it's amazing. Yeah.
yeah, I think like banding bush birds can be far better for the skin of your hands. I think. Are you planning to stick around in Australia?
I'd love to, but I'll just have to see where I'll find a job now. So I still have a contract with my research group at the moment until the end of the year. I'm still doing disease research, but then I'll just have to see what I find. I'm just applying everywhere, and I really hope I can stay. I love it here. I love the birds. There are way more parrots than in Germany. um, and yeah, it's just great here, and I hope I can find something that would be awesome.
That's great. Well, let's switch into the fun half, even though it's nowhere near half of the podcast. Let's work out whereabouts on the spectrum, on the bird nerd spectrum, you sit. Um, Now, having been a European birder and an Australian birder, what's tell us about the different field guides that you go to as a matter of choice um, in the different hemispheres.
Well, I think the European one's quite easy. That's obviously the Svensson and no other, which is a fantastic book. And here I couldn't decide. I arrived, I got different advice, and so just went all out and got I think the Piscean Night app for my phone, and then the Simpson Day as a book. And then I also have a Parrots of the World and yeah,
The
several parrot books as well.
the Joseph Forshaw Bible, the Parrots of the World.
Yep.
Yeah, isn't that a ripper? Isn't that an absolute ripper?
Oh, it's so beautiful. Oh, the paintings in there. My goodness.
I think I had one a copy of that from about the time I was 10 or 11, I think. So a long time. Maybe a fraction later, maybe 13 or 14. Um, but the only book I've ever had with birds that rivaled it in terms of my absolute wonder as I flicked through it was a sort of photographic study of waterfowl, and I think it was just called Waterfowl. And I don't have it anymore, and I remember I think it was published in conjunction with the Audubon Society, but it was magnificent. And those two books, when I was younger, just thought, oh wow, look at all these things out there in the world, these amazing things out there. So anyway, I'm stealing your thunder. When you're bird watching, because I'm assuming that you do a lot of bird watching because you describe yourself as an ornithologist, which would tend to sort of that would marry up pretty nicely. It's pretty logical. Do you have a favourite piece of equipment?
Just my binos, really.
Yeah, binos.
Because I usually carry a big camera as well, so I love to have a scope, but I just can't carry all these things. So I'm very happy with my binos. I currently have a set of Leicas, which are actually the universities, and I'm already terrified of having to return them at the end of my contract. So
Hey,
I'm already saving up to get my own pair. They're great, and I have them with me all the time. Yeah.
that's good. So, are, are they a standard pair or are they a small pair?
Yeah, standard pair, 10 times magnifying, and which is my favorite. I prefer the 10 times over the 8 times. Before the Leicas back in Germany, I had a pair of Zeiss, also 10 times magnifying. I think 10 by 42, and they're just great.
You're the you're the first person who's actually stipulated the magnification powers and the field that they use. So I like that. See, we're geeking out. We are geeking out. Um, have you got a favourite place? And again, you're familiar with the two hemispheres. So have you got different places? What's your favourite place to go birding? And this is birding for pleasure, not birding for work.
Oh, difficult. At the moment, just really near my house here. Just the bay near Geelong, Melbourne is just so great. So I love the Western Treatment Plant, of course, one of the just best spots here. The water treatment plant has such a variety. I think it's considered the best birding spot in Victoria. And that's very close to where I live. So I try and spend a lot of time there. Um, and just so the problem with the issue with the treatment plant is you can't just go in, you need a permit. So the easier option is just to go here at the coast in the neighborhood. There's a Ramsar site near here as well where I can see so many species in an hour and a half. So that's my go-to, especially during lockdown, where we can't really move around a lot. Yeah, but there's so many good spots, especially here in Victoria, with so many different habitats. But there are great places everywhere. I used to work in Austria, for example, for a while. I used to work with ravens, and that's another fantastic spot for bird watching. But now it's just pretty unreachable at the moment. Of course, even more so than with normal flight traffic. So I'll stick to the Western treatment plan for that.
But when you're in Europe, where would be your number one spot in Europe?
Depends on the time of the year, but one great spot is Lake Constance, which is one of the biggest lakes in Central Europe, one of the biggest drinking water reservoirs. And it's huge. When you're in the middle of the lake, you can't see the shores. It goes, I think, to 40 meters deep. People go diving in there. And you get a lot of seabirds there, even jaegers in winter. You get amazing amount of ducks and just water birds and shorebirds. So especially in winter, that's just absolutely fantastic.
And no renegade introduced parrots there yet?
Pardon, sorry?
Are there any renegade introduced parrots along the shores of the lake?
No, for them it's too cold down there. They're just mainly in the cities. They're so close to the Alps that just gets too rough for parrots, I think. With climate change, we'll have to see, maybe in a few years.
Yeah, maybe so. Maybe so. So when you go bird watching, is it an immersive experience for you? Are you just enjoying being out there, or are you madly ticking off species on your list?
Probably both. I love just being out there. I get completely immersed in it, especially also in just taking photos and just watching the behavior. So I don't just run around twitching, but I really enjoy just watching the birds for a while. But then I also record the species. The eBird app is just fantastic for that, because then all the birds you see already go into the global database straight away and can be used for research, which is fantastic. And I also then write the species I've seen down in my little book at home. So a bit of everything.
So, big challenge here. What's your life list number?
I don't actually know, which is shocking, but because for a long time I just wrote date first seen into my bird books. So I probably should start an Excel table or something, but that may be a bit too geeky. And also my European bird book is still back in Europe, so I can't actually count it all up at the moment.
Well, you're not at the extreme end of the spectrum yet. Nobody, not one guest yet has been able to tell me their life list number, and that's good because I just think that's a good place to be. If you can't just crack it off the top of your head, I think that's pretty safe. Now,
it's a challenge, though. Now I'm really tempted to count it all up. But,
yeah, I know what you're doing this afternoon. um, now, I'm guessing that it's going to be a parrot, but what's your favourite bird?
I don't have a favorite bird. There are just so many awesome ones. Love my Crimson Rosellas here. Love the ravens as well, my Amazons, and just the little robins as well. I just love the diversity.
Oh, you can't even come up with it. Well, a top three. You've got Crimson Rosellas, you've got the Amazons, the renegade invader Amazons, and which ravens are they?
Common raven, Corvus corax,
Just common
which are
raven.
spread over most of the world, and they are just so they're a lot bigger than our ravens here. They're just huge.
Yeah.
They occur over most of the northern hemisphere, and they're just fascinating. They're beautiful and they're extremely smart, which has just always absolutely flabbergasted me what they can do. I did some experiments with them and how they can plan ahead and these things, and it's just amazing. They're considered the apes of the bird world.
The apes of the bird world. Yeah. 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 No. Yeah. I hope that's because of their extreme intelligence and their use of tools.
Yes.
Yeah. Very cool. Very cool. Now, is there somewhere that you have on your bucket list for going birding?
To start with, definitely Western Australia. Haven't been out west yet, So I've been trying to explore as much of the continent as I could during the last 4 years, but being busy with the PhD, there's still some massive white spots on my map. So WA would probably be the first spot, and also seeing more of the center. Internationally, I'd love to have a closer look at Central America, find more of the Amazons in the original habitat, these kinds of things. And the biodiversity is just amazing. Small countries like Costa Rica or Ecuador that just have so many bird species that I'd love to see that.
Yes, I think we've all got Central America, South America on our list. Yeah, and you've got to get up the north of Western Australia as well as the southwest corner. Um, pretty amazing, pretty amazing. What's your bucket list bird?
Cool. At the moment, it's mainly just to tick off most, if not all, parrot species in Australia while I'm here. With on top of the list, maybe the palm cockatoo.
Yeah,
That's very high up the list, but at the moment, it's just impossible to get up there.
yeah. Oh, hopefully not. Hopefully not long. Hopefully not long.
Hopefully, yeah.
And
And I was,
we'll be out of lockdown again. Hopefully.
yeah, and then it would just be a road trip up Cape York. That would be amazing. I was lucky enough to spend about half a day up on Cape York doing another road trip where I just forced myself up there and had a look, and I was super chuffed to find some cassowaries. That was amazing. But I'm still missing quite a few of the parrots that are occurring even further north.
Yeah. Well, good luck with the fig parrots. They're pretty hard to find.
Yeah.
Yeah. Well, well, I think you've pretty much told us what your next step is, hopefully, and that's finding another position, another project here in Australia. But let's look a little bit further. Where do you want to? Where are you hoping that your career takes you in the sort of next, let's say, 5 to 10 years?
I mainly hope that I can just keep working with endangered species and especially endangered bird conservation in one way or the other. I'd love to keep doing research, but it can also be something more applied. Happily, I love field work and I've got a lot of experience with that, while also having the lab experience. So I really hope that I can just stick with that. And then apart from that, I'll try to stay a bit flexible because it's just really, really hard to find something. It's hard to plan really ahead, especially in the current climate.
Yeah, we've all just got to sort of sit down, wait and see, hope for the best. So
And do a bit of bird watching on the side, yeah.
exactly, don't waste your time doing nothing. Go out and get acquainted with your local bird population.